Hi, welcome everyone, wherever you are joining us from, to the second Global IMMUNOTALK of 2024. My name is Caetano Reis Souza, and I'm delighted to be part of this wonderful initiative, this brainchild of Carla Rothlin and Elena Zuniga. It has been running since 2020, and it benefits and inspires immunologists all around the world in an egalitarian and environmentally friendly manner. So it's a wonderful initiative. We have had some fabulous speakers over the years, and today is no exception. And it's my great privilege and honor to introduce today's speaker, Kodi Ravichandran. Ravi is originally from Chennai in India, where he actually started his career as a veterinarian. He went to vet school in Chennai, but then moved to the United States for a PhD at UMass Amherst, where he studied B cells, and then moved from there to Dana-Farber in Boston, where he did a postdoc with Stephen Burakov, working on signaling in T cells. And from Boston, he then moved to the University of Virginia in Charlottesville in 1996, where he started his independent career. And after 26 years at UVA, he then moved to WashU in St. Louis in 2022, where he is currently the Robert Kroc Professor and Chief of the Division of Immunobiology in the Department of Pathology and Immunology. And I believe that Ravi also has an additional outpost in Ghent in Belgium. Now, Ravi actually abandoned his focus on B and T cells when he started his independent career, and really decided to focus primarily on this fascinating process of efferocytosis, which he will tell us all about today. And really, he has made outstanding contributions to that field by defining find me and eat me signals emanating from apoptotic cells, as well as phagocytic receptors that mediate that uptake both in professional and non-professional phagocytes. And importantly, he has really put efferocytosis at center stage of both homeostasis and pathology by showing how impairments of efferocytosis can lead to pathology, and conversely, how increasing efferocytosis can be harnessed as a therapeutic strategy. And for me, really, it's the clarity of Ravi's thought and discourse that is a key characteristic of his research. Is very patent in many elegant review articles, and in particular in many talks, as we will see today. So as usual, the talk today will be followed by a Q&A session via Twitter or X, in which Ravi will endeavor to answer your questions, and the instructions for how to participate in a Q&A session will be put up at the end of this talk. But so just before we let Ravi get started today, as usual in this series, I will start by asking him a more personal question for the talk, which is really Ravi, how would you describe your personal style, and how did it impact your scientific career?
It's a great question. First of all, thank you for inviting me, Gaetano, on this very nice introduction, and for everybody else who is joining us. I'll start off by saying that I was extraordinarily lucky to have mentors, both as a graduate student and as a postdoc, who taught me a couple of principles. And one is that you can do really good science, but you can still be a decent person. And so that sort of has helped me guide through that, both that happens in the lab in terms of respecting people and colleagues, as well as outside. And when you review papers and grants, and it's always, you can hopefully do very good science and do it in a high standard without being a jerk about it. And the second, I would say, is also, I try not to pick favorites in the lab. There are no favorites. However many people are in the lab, they're all my favorites. And so that's something I learned through family. And so that's something I've followed through that has also helped me. And the third thing is also, there is zero internal competition in the lab. Now, what that then fosters is a really open exchange between people in lab meetings and journal club. It's us versus the scientific problem rather than each of us against each other in any way. That is, I cannot tell you how lucky I've been over the years with the group I've had. So these have helped me move forward.
Well, it's a great testament to your mentoring, and thank you for sharing it with all of us joining from all around the world. These are important messages, I think, for everyone working in science. And so without further ado, Ravi, I'm going to ask you to start your presentation, and you've got this very provocative title, How to Eat a Good Apoptotic Meal, Lessons from Phagocytes. Ravi, the stage is yours.
Thank you, Kato. I hope everybody can see my slide, and if somebody could give me a thumbs up, that'd be great. Hopefully everybody can hear me okay as well. Great, thank you. Okay, what I wish to focus is I'm just going to give you a brief overview of the steps in apoptotic cell clearance process so everybody can be up to speed on this. And then I am going to actually talk about some tools we have developed for tracking apoptosis and efferocytosis in vivo. As I was just mentioning to Kaitono's question, over the years, I've been truly blessed in terms of the students and postdocs who have been fortunate enough to come to the laboratory and do some outstanding work. And I was thinking what to present. And for the large audience, what I decided to do was talk about tools that may be very generally applicable for a larger group of people, and that still tells us new insights on apoptosis and efferocytosis. And the second was, again, another tool we developed, but it helps us address how to modify efferocytosis in vivo. That's really the major focus. Before we go on to that particular problem, as we're in this conversation, we turn over roughly about 1 million cells a second in the body over the course of the day. These, if you did the math in another way, it's about 330 plus minus 20 billion cells per day as part of routine healthy living. In fact, I'm going to refer to this review, which has really done a wonderful job of putting this in perspective. Who are these cells that we turn over? These are excess cells. Many tissues make excess ones, bone marrow, especially thymus, for example, we know those. This is also true for in the brain during neurogenesis. Only the fit cells are allowed to move on, while the cells that are deemed not perfect enough to move forward, they're for whatever reason selected against. Then, of course, we have like cells such as used or aged cells. Most cells have a specific lifespan. Think neutrophils, think red blood cells. They all, after a certain period of time, they go in. Of course, you can also think of damaged cells when, for example, we constantly undergo damage in the context of in the skin due to UV light, or in the context of the eye every day as part of the photoreceptors undergoing light-induced damage. And of course, if you do the math, it's roughly like RBCs constitute about 2/3, and then the neutrophils are another roughly about 1/5, and the rest go around. Of course, the gut epithelial cells are huge as well. Actually, it's about 80 grams, which turns out to about 2 size eggs. I thought it was just a fascinating number. A very, very important thing that's relevant for today's talk is that majority of the homeostatic turnover happens via caspase-dependent apoptosis. As most of the people in this audience know, there are many different forms of cell death. There are multiple modalities of apoptosis, necroptosis, and other forms of death as well, that apoptosis that are induced in very specific circumstances. But the apoptosis, based on really nice work from Andreas Strasser and others, that is about 90+% happens via caspase-dependent apoptosis. And in fact, the cellular turnover I already referred to was a paper from Sender and Milo that I pulled out some of this information from. So the obvious question is, what happens to these dying cells? This, again, began thanks to some fantastic work by a number of people in the late 80s and early 90s, including Chris Haslett, John Saville, Peter Henson, a number of other people. I'm missing about 30 other people. So, but the one term that is now pretty much used widely in the field is now called efferocytosis to refer to this process of clearing apoptotic cells. This was coined by Peter Hansen approximately around 2002. And the word he coined because was Effero referring to carry to the grave, and the process being, instead of phagocytosis, it's an efferocytosis process of carrying dying cells to the grave. And the other aspect that also comes within is that many of these, in the context of, say, inflammation, there's even more cells that come to these tissues on top of the regular turnover that happens in all these tissues. What are the phagocytes that perform efferocytosis? We can immediately think of professional phagocytes, the macrophages, and the immature dendritic cells that are quite capable of eating debris and dying cells. Then, of course, something that we don't pay as much attention, but is now starting to get much better attention, is so-called non-professional phagocytes. The reason these are called professional, these are non-professional, is because simply the macrophages can just eat many more corpses in a much shorter time, and it can process the corpse load much better. Whereas the non-professional phagocytes, they may eat fewer of them and in a slightly slower kinetics, but there is a very important distinction. That is, these are numerically has a supremacy. There are way more epithelial cells, and they can eat their neighbor easier than a macrophage having to access it. So we shouldn't ignore them. It's kind of like the tortoise and the hare, that they may continue to do a heavy burden of clearance, but not at the same magnitude as a macrophage. In fact, there's evidence now that fibroblasts can eat, epithelial cells can eat, and even endothelial cells can eat. And then there are also specialized phagocytes. This is where, for example, Sertoli cells of the testes, which at any given time can be associated with about 40-ish spermatocytes and various stages of differentiation, but will specifically eat one or two that undergo poor meiosis or cannot move forward while still nursing all the others. They're just phenomenal cells. And then the same way is this retinal pigmented epithelial cells, or RPE cells in the eye, which again have every day in the roughly around dawn time, clear out all the photoreceptor outer segments in a beautiful way routinely every day. And so these are specialized cells, Sertoli cells on the RPE. We pretty much live, we are born with them and we die with the same number of them, yet they continue to do this throughout our lifespan. Now, I'm going to show this movie for a very important reason, because it captures all the different events that over the years work from a number of laboratories have defined. So for example, here is an apoptotic cell, which, it's a target cell, which is green, which is not dead yet. And then here is one that's starting to die and starting to look yellow. I'll explain these colors at a later point when we get to them. But for the moment, just think of them as this is a dying cell, this is live cells. Here's your macrophage. And you can see that this macrophage is going to get up and leave and go and find this. So let me play this again, but with a different thing. So in this case, this macrophage is now touching another macrophage, but it just quickly says, nope, you're not tasty enough, or go away. The other macrophages, then it starts to move. Now it bumps into another cell, which is fully forms a nice attachment, but this is a live cell. But the macrophage does not eat this one. And then it finds, now it actually senses that there is actually a corpse here, then it actually moves very quickly and actually eats this one. And you put in a certain probe that then progressively get red as the corpse is being processed inside the macrophage. I'm going to stop this movie to stop it from being disturbing, but the work from various labs, what I've identified is there's a very early stage, this macrophage can actually sense the presence of apoptotic cells. This happens via find me signals, which are released from the dying cell that provide an attraction cue for the macrophage or a phagocyte. And then, of course, there are don't eat me signals. You saw them, same macrophage touch live cells and then not engulf them. Then, of course, you have taste, which I consider as the ligand receptor interactions that provide the specificity for eating that's actually present in the phagocyte. And then, of course, the physical act of internalizing another corpse. And then lastly, the digestion step. This is basically like your a neighbor moving into your house with all the belongings and half of them broken. And sometimes these macrophages, as you will see in a few minutes, can eat multiple corpses. There's an enormous amount of metabolic load that's being acquired, which needs to be processed by the phagocyte. On top of it, contrary to other types of phagocytosis, say Efferocytosis phagocytosis or complement receptor phagocytosis or bacterial uptake, etc., the process of taking an apoptotic cell is largely, I say it with a cautious note, because there are some examples where it may not exactly happen this way, but it's largely anti-inflammatory. What I mean by that is this process, contrary to other types of phagocytosis, is potently secretes anti-inflammatory mediators and do not secrete pro-inflammatory mediators. And in fact, this is actually why my lab got into this about 20 years ago, because we were interested in what is unique about this process that's anti-inflammatory that led us to the process itself. The topic I want to talk about is something that the field as a whole has had a major challenge for, I would say, probably 15, 20 years now, since we started more and more about the process of efferocytosis. That is the real-time tracking of apoptosis and efferocytosis in vivo. As immunologists, we all know how millions of immature cells die in the thymus and bone marrow as part of development. Yet when you take sections of these tissues, it's usually very hard to detect dying cells of them, except in disease states where obviously the system is overwhelmed and we cannot see, and we really cannot see, until then we cannot really see apoptotic cells. Even then, when we actually score these, usually these are endpoints, as I would call them, which is basically cleaved caspase-3 staining or tunnel staining, or where the process has already happened. You don't really watch as it happens. And so about 4 or 5 years ago, my laboratory had a really lucky break, in the sense that we were very fortunate to, it's a graduate student by the name of Mike Raymond who joined our group. There are people who really have a bent in making tools, and Mike is one of them. And Mike set out to design a dual probe that can detect both apoptosis and efferocytosis in vivo. I don't want to bore you with all the different versions Mike attempted, except to say it required almost close to 100 different constructs that we had tested, back to the drawing board, testing again. And then it finally led to the design of this probe that Mike coined the term for Charon. So what is this probe? The way he did this was he would take a GFP and he would put a little linker in the middle. And this particular linker, when it's cleaved by apoptotic caspases, now this GFP becomes positive. There are a number of issues with the regular GFP, and that being that many of them are very quickly quenched in the acidic pH, even very low acidic pH. So he had to make it more stable. Then he went back into the literature on various designs of GFP, and then he went back and made a lot of mutations that made it more mammalian specific. It required a significant amount of engineering that led to the design of this, what we now call as pH-Cas GFP. What this is this is expressed on these cells, and when there's no apoptosis, meaning in this case it's a UV signal, there's no signal for GFP. But now when we give a UV trigger to induce apoptosis, and this is about 4 hours later, you get about 2/3 of the cells beautifully becoming positive for Cas GFP, and also the annexin-5 positive now as an indicator on the cell surface, which is the exposure of phosphatidylserine on the outer leaflet. This is specific for apoptosis because if you add this small molecule called QVD, which is a pan-caspase inhibitor, and then what happens is the signal, which you see very nicely, goes away. There's another important thing here. Here's a UV trigger, is an ABT, which is another way of killing cells, or staurosporin. All of them induce apoptosis, where you see a nice signal. In every case, the signal goes away. So then what Mike came up with was he wanted to design something, this will tell us apoptosis, but he wanted to also design what will be possible to monitor efferocytosis. This happened because we were extremely lucky that the Campbell's group in Canada, which makes a lot of these probes, were extremely lucky, extremely generous in sharing with us and some unpublished reagents, and especially this pH sensor. What this pH sensor is, it has a low fluorescence at neutral pH, but in low pH, it becomes bright red. And so this allowed us to monitor apoptosis via the pH-Cas GFP, and then via this probe, which we call it Florina, can actually monitor the apoptotic corpses. And so this is what made Mike to call this caspase and pH activated reporter, which turns on. And this is in part a nod to Charon or Haran, which is in Greek mythology, the person who carries the dead across the river Styx. And the way this works, supposed to work, so Mike made a probe that will have pH Cas3P with the T2A sequence in between, and then fluorina, is that as the cells undergo apoptosis, they'll be eaten, and then the green will start to die as soon as it goes to pH 5-ish, and then the red will start to arise. So you will have initially green, and then the handoff to red, basically. And this is exactly what happens. Here's an in vitro experiment where he's now adding macrophages with these apoptotic cells. These apoptotic cells are now beginning to be eaten. And so you can say that the green has gone away. And as the green goes away, the red starts to appear within the same macrophages. In fact, you can score this very nicely by various modalities. And so the fluorina signal goes up while the Cas3P goes up. And in the engulfed cells, basically, there's a greater ratio of red over green. And this works actually in a live imaging. So here's a green cell that's eaten, and now it turns red beautifully inside after it's eaten. I'm going to stop this movie and then show you something else. Mike also asked, would this allow us to tell multiple corpse uptake? So here is a macrophage that's eating 3 different corpses that we have loaded with, and then you can watch this particular one. Here in this case, it's a movie that shows that, the image that shows that, but here we can see that this one macrophage is now going to eat 3 different corpses that are nearby, and each of them progressively becoming red as they go along. And so this tells us that you can actually eat, that we can monitor multiple corpse uptake. As we were working on this, we were very lucky to have a fantastic collaboration with Will Wood, University of Edinburgh. Will has been a longtime friend, and then he recruited an incredibly talented postdoctoral fellow at the time, Andrew Davidson, who now has become a faculty at University of Glasgow. And this led to a really, really productive collaboration between the 2 labs that helped us identify some key features of macrophage eating in vivo, because we're still in the process of making mice and characterizing them, but this gave us a quick window to how to actually study them in vivo. So the way we did this was he would make pH Cas3P and fluorina, and then this was expressed in Drosophila. And in the context of Drosophila in the nervous system, what happens is that there is glia that sits here, and then below them is the hemocoel, where these hemocoel macrophages, they actually are hemocytes that actually move around underneath them. And so you can actually monitor both of them. And in fact, this is a quick shot at this. You can see this like a Christmas tree look, where these green cells are your dying cells, and the red ones are the ones that are taken up different, they are taken up by the various phagocytes. And when we go back and quantify them, both the glia and the hemocytes, or the hemocyte macrophages, they both eat. But the ability of the glia to eat is much less because they're not very motile. They just sit there but send out the projections. Whereas these guys move around, as I'll show you in a second, and therefore they could actually eat much better. So we focused on the macrophages. So some insights from watching and quantifying macrophage efferocytosis. The first one is the macrophage has an ability to rapidly clear a huge lawn of cells in vivo. And so in this particular case, here is the head and here is the posterior part. And these macrophages are also labeled green here, but you should still be able to tell the macrophage from the cells that are dying, that are bands of cells that are going to start to appear as green. And then some of them will become red because they're being eaten. You may wonder, what are these reds already? That's because they're in the lower plane. There are macrophages still underneath the plane in which we're now visualizing. And so as you can see, these macrophages start to move rapidly, and they start, you can see all these apoptotic cells are emerging. These macrophages literally gorge on them as they go along. Once they begin to eat, you can also see them acquiring green first, and then they become progressively red inside the macrophage. And we can actually track essentially every single macrophage in different planes, quantify them, how many corpses are in individual macrophages over a time course. Easy for me to say, but it was really done by Andrew Davidson and Mike Raymond collectively. So the point was, a macrophage is really rapidly clear a lawn of cells. this also gave us the hint that maybe that's a partial explanation for why we hardly see apoptotic cells in vivo, because they're very quickly removed. This also led us, as we were doing this, the macrophages sort of follow the strategy of eat first and then digest later. What do I mean by that? So here is a movie in which we have now taken individual images at different times of a single macrophage here. You can see here's a macrophage that's just starting to touch an apoptotic cell. In 1 minute, it's touching. In 2 minutes, the corpse is already inside the macrophage. Now, if you keep tracking them, it's around 20 minutes, like 17 to 20 minutes, that when the corpse starts to become acidic because of the red, the probe we have put in. So what this tells us is that actually the eating is not the rate limiting step. It's a degradation of acidification that's a rate limiting step, which also explains how the macrophages can successively eat multiple corpses, then take their own time to eat. Mike jokingly said to me that it's like we don't stop after eating an appetizer when we are in a restaurant or when we're eating dinner, and then we go on to the main meal, we go on to the dessert, and then come home and digest them. It's not that different. That's what Mike jokingly said to me. And in fact, you can quantitate this. Now, if you go back and look at the mean corpse burden, this guy progressively goes up. Around by stage 14 of the embryonic stage, you already have maximum, essentially, of corpse numbers. Yet when you look at the acidification, it's much delayed. In fact, it's 15, it starts to begin. It's really at the stage 16 that the maximum acidification happens. The other thing which we also noticed was even macrophages that have a significant number of corpses in them move around quite readily. So here's an example of one of them. So you can see some of these guys who with chock full of corpses, they still move around and they eat more corpses. And there's nothing that's really stopping them. I'm going to play it again so you can see them moving around quite readily and eating additional corpses. In fact, this was quite impressive how they could continue to eat even after eating multiple corpses. There was another thing we also noticed very quickly. It comes as no surprise as macrophage heterogeneity, but this macrophage heterogeneity is also reflected in how well these macrophages eat. And here's an example of one of them. Here is a macrophage which is absolutely chock full of corpses. I believe it had close to 18 or so. And here's one which had just 1 or 2. In fact, we can go back and quantitate each of them. And so we call them low burden and extreme burden corpses. So you can see, quantitate them. And if you see at stage 12, most of them are 1 to 3 corpses. And then a few have 4 to 6. Then as you go progressively, there are in later stages, you have 10 plus corpses in some of them. And the number of them is 1 to 2. There are still a few with just 1 to 3 and 4 to 6. So there's a full distribution that happens, even though essentially all of these macrophages had access to the same number of corpses. We have also seen them in vitro. And so we kind of think about them as these Joey Chestnut type macrophages that can eat a lot of corpses. This is a question that's in the lab as to what determines how certain macrophages can eat much more. So this paper came out in 2022. We have since made the Sharon mice and the Sharoff mice, they're still being analyzed as we speak. This work also raised some interesting questions. How can a macrophage eat so many corpses so quickly and also digest them? Could eating one corpse help eat the next one? If so, would there be rapid transcriptional changes that facilitate continued clearance in a macrophage? I'm happy to say that Turan Tufan in the lab has done some really nice work along with Gamze Kumarpe. And so both of them have defined certain steps of what dictates these eating as well as the transcriptional programs that happen in these high eating macrophages. You can also see this in the context of in vitro. Here is an imaging that was done by Chanchal Chowdhury in the lab. And you can see that here's a macrophage that has a single corpse, and here's a macrophage with multiple corpses literally next to each other. And this tells you that they have an ability to eat multiple corpses. And then obviously then raises the question of, how do you study this? How do you study it in vitro context even to figure out the responses? In fact, one of the challenges, and that was actually that we all faced until very recently, was Most of the efferocytosis assays, what you do is you take an apoptotic cell separately, be it your thymocytes, be that your Jurkat cells or some other cell type of your interest, you induce apoptosis, then you label it, you wash it off, then you feed it to a macrophage. In the process, you have removed many of the apoptotic cell factors, which we talked about as the find me signals, etc., which also program the macrophage. There are a number of other features that are removed, and then we mix the two of them, and then we look at the response. My lab has also been doing this until very, very recently, and we still do that as well, because it has some very specific advantages to that. But one of the things that we also wanted to see was, can we capture the macrophage, engage apoptotic cells in sort of a pseudo-native environment? I.e., can you still keep the find me signals, the contact, the eating, and the processing? This led to our second generation probe, which is now called Sharaf, which is also designed by Mike Raymond. Basically, the Sharaf now has a slightly different flavor in the sense that now the red is caspase induced. So the CASP RFP is a probe that when there's a caspase 3-7 activation, this becomes red. Then there's a second probe we put in. It's called SEP, which is one version of GFP, which is a pH sensor. In this case, what happens is it's basally green, but once it's eaten, what happens on low pH is that the GFP gets really, really quickly quenched. And so the idea is that you would start off with the cell that's apoptotic cell that's green and red. And then once it's internalized and digested, now you only have red, but the green would be gone because the GFP is quenched. The one huge advantage of this probe was that Sharon, while it worked nicely in flow cytometry, it was whereas this one works much more easily and is so much better suited for flow cytometry. And this is just a proof of principle that you can see that the CAS GFP beautifully happens in terms of the red. And this is when you add QVD to inhibit apoptosis, the red signal does not arise. And so Mike made this CASP RFP with the T2A site and the SEP. And then when you go and do this, Sharaf detects efferocytosis because no apoptotic cells and plus apoptotic cells, the RFP is definitely there inside the phagocyte. This RFP does not get quenched in the phagocyte, which is actually good. Then when you go back to the Sharaf GFP signal, when you have normal conditions, while this is red, the green is largely gone on most of the cells. But yet, if you were to add something like baphyllomycin that prevents acidification of the phagosomes, then the green signal still remains. This is 80% of this 26%. So all of them basically still retain the green. So that tells you that it's really a caspase dependent, as well as the GFP pH dependent response to ecstasy. This then led Mike to do what we now call as a one-dish efferocytosis assay. And he puts in another one, which is called this inducible caspase 9 into the same probe. Now you basically transfect your cells with them. So the fibroblasts are now transfected with the macrophages in them. You selectively kill the target cells. And so they undergo apoptosis, but then in the same dish, you have all your find me signals. The macrophages move around, they eat, and then you can also study the responses. And because you have the colors in them, you can actually pull them out and then do your analysis as well. And here's an example of these fibroblasts dying beautifully in this context, and then even in the one dish assay can very nicely detect the efferocytosis by these macrophages. And one other thing that Mike came up with was, can you then engineer additional probes such that you have different colored corpuscles in the same dish? So here's, for example, one is with the CASP RFP. There's another one that's called as my RFP 720. They're both induced to die by caspase 9. Then when you do them, now you can actually score the same macrophage eating one corpse, just the red, or one carrying only the purple, or red and purple, which would then tell us how they are actually there. In fact, that's exactly what's shown here. Here is 14% of them macrophages have the red, about 16% of them have the purple, and then a subfraction of them, about a third of these have the purple, and a third of them have, 1/4 of them have the red. The key point to take away from this is not 100% of them have both, which then again tells you that some of them do not have the second corpse at all. What dictates some to eat more and some to not eat more? And then we can also go back and look at how many of red and how many of purple are per cell. This is actually ongoing. It has really given us a nice way to tell what factors control continuous efferocytosis. The reason for this is when you have any kind of an inflammatory context in vivo, it's not just one, it's often a macrophage has to confront multiple corpses. Therefore, it has to successively eat multiple corpses, which is really important for clearing this large number of dying cells in the tissue. And this actually allows us to address those kinds of questions. So now I'm going to switch for the next 20-ish minutes to a different topic, and not a different topic, but an extension of the topic. It's well known that there are errors in efferocytosis. And this can happen in one of two ways. Either we have deficiencies in specific receptors, we have other deficiencies in the machinery that eats, or we are overwhelmed by a large number of corpses that arise quickly in a local tissue. In all of these cases, it's known now there are multiple examples. Inflammatory diseases, in atherosclerosis, Ira Tabas has done a really nice job. There's acute kidney injury cases where it's known. There are retinal diseases where this is a problem. And then, of course, there is liver toxicity as well as arthritis, where there's been linkages to failed clearance of apoptotic cells, and this in turn leading to the corpses become secondarily necrotic, and that in turn providing a milieu that's pro-inflammatory. The question that we asked a few years ago was, is there a way to boost clearance of dying cells for debris and thereby improve outcomes in inflammatory diseases? For this, we needed to go back a bit in time. Many years ago, around 2001, my lab identified this protein called ELMO, which is a homologue of CET12 in C. elegans. It was a fantastic collaboration with Michael Hengartner's group at that time, between my group and his. We identified this protein, which we named engulfment and cell motility, or ELMO. ELMO was not a, it was mostly an adapter protein, had a bunch of armadillo repeats, had a pH domain and binding, some other binding sites, but it's important for engulfment. And ELMO-NEL macrophages have a partial defect in eating. CET12 deficient worms have a defect in eating. And in fact, what's nice is that you can take the mammalian or human ELMO, put it into the CET12 deficient worms, and rescue some of the defects in these particular worms. So ELMO functioned upstream of RAC, but we didn't know how initially. And then some work from a number of people in the lab over the years, we went on to show that ELMO binds to another protein called DOC, and then to another protein called CRAC that it binds. And this complex works as a guanine nucleotide exchange factor for RAC and converts RAC from the GDP state to a GTP state. We still don't know what the upstream receptor was. This was some very nice work done by Daeho Park in the lab, who identified one protein as possibly upstream of ELMO, and that is actually a protein called BAA1, or brain angiogenesis inhibitor 1. This has 5 thrombospondin repeats that directly engage phosphatidylserine. It's a 7 transmembrane protein and is a type 2 adhesion receptor GPCR family member, which has a large extracellular domain that can mediate these, as well as a long intercellular tail to which ELMO binds. And then the sound goes downstream. BAA1 is expressed in most cell types, in barely detectable levels in macrophages, but largely expressed at a very high level in non-professional phagocytes. And in fact, we could actually see this particular slide I put in for a couple of reasons. One is, besides BAA1, there are a number of other receptors work from a number of groups. So Lemke and Carla has really shown us very beautifully that a TAM receptor family, which via Gas6 can actually bind phosphatidylserine indirectly and thereby signal downstream to mediate responses. There is Nagata's group identified both TIM4 as well as Ray Burgess' group, this recognition of MFGE8 through integrins, that both of them then engage. TIM4 directly engages PS, but has no signaling motifs, whereas integrins, of course, are well known to signal through through other players. And then, of course, the BAA1, which is a 7-transmembrane protein that can engage, but can also directly engage the downstream signaling players to lead to RAC-GTP activation. Of course, the RAC-GTP has multiple roles. It obviously involved in actin polymerization to facilitate the formation of the phagocytic cup, but it's also involved in anti-inflammatory signaling pathways that work initially from Peter Hansen and Valerie Faydock, but subsequently by others have all defined this. This work was done by Sho Moriyoka in the lab, and he was a postdoctoral fellow. Now he's an independent investigator at University of Virginia. He's doing really well in his own lab. And when Sho started the lab, one of the questions we had was, we have been making transgenic mice with BAY1 directly. They were okay. They did some things okay. They slightly boosted efferocytosis, but they were never very strong. This one, Sho and I designed this particular probe, where this is basically a Belmo. We call them Belmo because this is BAY1, where we're truncated it here, then directly fuse the Elmo. And then this would then cut out the middleman, so it can directly now recruit DOCK and increase. First of all, does it get expressed? And the nice thing is the Belmo nicely get expressed on the cell surface. And importantly, it eats really nicely compared to the control eating where we have a shorter time frame. It's only about 20% of the phagocytes are eating. And here they boost it to about 60% of the cells now begin to eat. And it's not just the number of cells eating. On a per cell basis, they have many, many more corpses in them. And they don't eat live cells. So it's really PS dependent eating. And this is also sensitive to annexin 5. And so it's inhibited by blocking the phosphatidylserine, as you would expect for Elmo. And you can also do this another way, which is you make the Elmo mutated such that it cannot signal downstream. And here's your Belmo 6M, or a mutant. It's essentially like the control, but while the Belmo continues to eat. And interestingly, it's a progressively continuous eating that increases. Now, when you go and look at the individual cells, the mean fluorescence intensity of them is also quite high. So Sho went on to make a transgenic mouse, and that is put into the Rosa locus with this lock-stop locks, and then it's an iris and then an EGFP. We crossed it to a number of different CREs, CXCR1 CRE to target macrophages, Villan CRE to target epithelial cells of the gut, albumin CRE to target hepatocytes, and PepsiK CRE to target the kidney epithelial cells, proximal tubular epithelial cells. And then we make the tissue-specific Elmo, and then where the Belmo expression happens, and then we can check. As a proof of principle, we just use this particular one here. It's a CX3CR1 CRE Belmo, where when we inject apoptotic cells into the peritoneum and then isolate the macrophages 20, 30 minutes later and ask how are they, so they eat much better than the control macrophages in the same peritoneal lavage. Based on a number of other data, what we found was actually improving efferocytosis by non-professional phagocytes, such as your epithelial cells and other cell types, or hepatocytes, may have a much better effect on disease outcomes. And therefore, we tested Belmo expression in several models targeting these cells. To say it in another way, and perhaps as a joke, giving, say, $100 to Jeff Bezos or Elon Musk may not make a huge difference, but yet when we do this for somebody who may want this money, this can actually make a difference. And because it's non-professional phagocytes generally tend to eat less proficiently, this actually can make a difference. In fact, this was the case. I'm going to zip through 3 different models. All of them essentially show the same thing, which is here's a Villan CRE Belmo model. Then we test them in a DSS colitis model. You can see a significant inflammation here, which is much better improved in the Villan CRE Belmo context. And here's the histologically, can actually score them as well. The Villan CRE Belmo expressing mice have a much less effect. And in fact, when you go back and look at the corpse burden, the tonal staining, you can see all these tonal positive cells in the control mice treated with DSS, whereas in the Villan CRE Belmo, there are a few, but they are substantially reduced compared to them. And this is the tonal positive cells. They've also done the exact same thing with cleaved caspase 3, and the answer is the same. There's one other thing that Sho noticed was that when you go back and looked at the cytokines in them, in fact, there was a much more TNF in the control mice compared to the Belmo-expressing mice, which substantially reduced this. The same with IL-16 and MCP1. On the other hand, the IL-10, which is known to be an anti-inflammatory molecule in some of the responses, and particularly in intestinal tissue, this now has a nice increase in the Belmo case compared to the control. We can also do the same with albumin-Cre, and here you actually add a molecule called DEN, which induces hepatocyte toxicity, and you can see in the control mice there are a significant number of terminal positive cells, but it's substantially reduced in the Belmo cells. And here is, I'll come to that point in a second. There's a very trivial explanation that could happen. What if these Belmo mice do not undergo apoptosis at all? I don't have the time to show you all that data, but this is something that bothered us quite a bit. And we can almost say in every case that the initial onset of disease is comparable in both of them. And in the initial window, the number of dying cells are very comparable. Just in the latter stages, they just get cleared better. And in this case, there's another experiment that showed it was you could actually measure the DNase2 activity, and normally the DNase2 is inside the macrophage phagolysosomes, and that's where the DNase2 actually chops up the DNA that comes in from them. This really nice work from Nagata's group has shown this. And again, what you find is that when you score for this, there's much more DNase2 activity in the albumin-Cre Belmo context compared to the albumin-Cre control context. There's no Belmo expression. He also did this in another model, which is a cisplatin model for kidney injury, and the answer is the same. There are more dying cells in the pepsi-K-Cre control, whereas the pepsi-K-Cre driving Belmo, you actually have much less. And similarly, you have more cells eating, and in turn, these are the efferocytic phagocytes in the kidney tissue. You see much more, and this is actually scored. And this kit that we use is called the apoptotic. And you can also show this by creatinine levels, which are much higher in the pepsi-K-Cre control conditions compared to the pepsi-K-Cre Belmo condition. Also, in terms of when you give a lethal dose of this, the Belmo mice survive so much better than the control mice, which progressively die by about 6 days. I think this is the important control I wanted to point out, that the transgenic mice undergo early disease onset, indistinguishable from the control mice in all of these models. Obviously, what is the mechanism behind this? And in the interest of time, just going to shrink it to a very simple message, and that is majority of the genes that are upregulated, or one of the major genes that are upregulated, are actually so-called proteostasis genes. In other words, an ability of a macrophage to manage excess protein load within them. And so, for example, some of the ones that come up are ER protein folding genes, much more upregulated. And then these are degradation of unfolded protein that also happens. Then other ER functions, they all go up. I do want to say something is that not everything is absent here in different time points. You do get some of them here as well. And then this is with the apoptotic cells, they're just more. So in fact, if you actually block BIP, which is one of the molecules that are involved in protein folding, this effect, this increased eating between here and here that you see with the Belmo versus control is basically decreased in the context of when you add BIP siRNA. The same with DNase3, ATP2A, VIMP. Every single case, this effect is partially blunted back to almost the control stage when the proteostasis genes are actually affected. This is just the control. We did it by shRNA or by using CRISPR-Cas-mediated knockdown, and the answer is very, very analogous. If you say if it's proteostasis related, can you actually measure it in the kidney injury model? And here again, the Belmo was much more protective when you do this ischemia reperfusion injury model, or IRI. And again, compared to the control condition, the Belmo mice are much, much more protected. This is also reflected in their viability. When you go back and do histology, I hope it comes through on your screen. It's compared to the necrotic area in the control condition, the Belmo condition, there's much less necrotic tissue. At the same time, if you take the sham D2, they're very similar in both mice. What about ongoing disease? Because if you ever want to use it as a therapeutic approach, one would have to do it in an ongoing disease. This is where we faced a technical problem that Show beautifully overcame, and that was the BAY1 is a very large molecule. And then on top of it, if you add an LMO, packaging that gene became tricky. Therefore, what Show did was he took TIM4, then he chopped off the tail of TIM4 and directly put LMO1 into it. Now he called it TELMO. And again, it's nicely expressed on the cell surface. And just as in the case of the BELMO, it's very nicely increasing efferocytosis. This is sensitive when you get rid of the signaling via TIM4, LMO, or interestingly, when you just express TIM4, that also boosts because it's known, and you don't need the tail for it. But then when you have TIM4 that cannot bind phosphatidylserine, you lost this effect. So it basically goes from here back to here. Now, if you take off, so what Show did was he basically generated adeno-associated viruses or AAV-based introduction of TELMO. The idea is that one would inject it on the left renal vein of the mouse. Then you do ischemia reperfusion injury, you wait for a few days, you do ischemia reperfusion injury on the left, then wait for 14 days, and then assess this. The left kidney is usually is the one, but you actually remove the normal contralateral kidney because most of the mice can survive with just one kidney. Now what happens is the mouse has to entirely rely on the left kidney. Then you ask, how do you evaluate the function? And in fact, what was really pleasing was compared to the vehicle-treated condition, this is BUN on the left, the TELMO mice were much better. And this is the no nephrectomy case. So almost at the level of no nephrectomy. Here's a fibrosis staining. Here's a fibrosis staining in the control mice that went through IRI. But here is the where we have expressed TELMO after day 2, how this would actually happen. And here's your AAV TELMO. This is the vehicle condition. And here's AAV TELMO that improves it much better. So compared to this condition, this is much better. This is again scored here in terms of VIP expression, which is one of the proteostasis genes that are relevant. So the basic message from this is that chimeric efferocytic receptors, such as, which we call as chefs, such as BELMO or TELMO, promotes enhanced efferocytosis with anti-inflammatory signaling and dampens inflammation in a different inflammatory disease context. Show spent a significant amount of time making cover image for cell, which unfortunately was not picked, but this was actually made by an innate person in it's called Cells at Work. Here is the acknowledgement to them that they actually made. And this basically shows here is a cell that's actually eating, supposed to be. So now I finally want to acknowledge the people who actually did the work. The first part of the work was done by Mike Raymond, who is now in Boston University as a postdoctoral fellow, and he's now starting to look for his own jobs very soon. And Sho Morioka, who is now an independent investigator, and he did the initial work as a postdoctoral fellow in my lab, and he's continuing this work as well. And as Cayetano already mentioned, I'm extremely happy to have another group in Ghent that's also there. These are the various funding sources and various collaborators as well. And thank you for the opportunity to present this work.
Well, thank you very much, Ravi, for a fascinating talk. I just want to remind the audience that there will be an opportunity to ask questions via Twitter. The instructions have been put up on the screen. And Ravi will do his very best to answer them in the next few days or weeks. But so it just rests for me to thank him once again for making time today and sharing his wonderful science with us. And don't forget to join us next week when we have the talk by Maria Mata. So to all of you out there throughout the world, enjoy the rest of your day. And thank you again for joining the Global IMMUNOTALKS community.
Thank you, everybody. And thank you, Cayetano.